अस्सलाम वालेकुम, वेलकम टू माय चैनल। आज जो हम एक्सपेरिमेंट या प्रैक्टिकल करने वाले हैं वो है प्रिपरेशन ऑफ ह्यूमन मेटाफेज क्रोमोसोम फ्रॉम ब्लड लिम्फोसाइट। मतलब जो हमारे पास ह्यूमन मेटाफेज क्रोमोसोम है उसको हम ब्लड लिम्फोसाइट से कैसे सेपरेट करते हैं या प्रिपेयर करते हैं, ये हम प्रैक्टिकल करने वाले हैं। ऑब्जेक्टिव: कल्चरिंग ऑफ ह्यूमन ब्लड लिम्फोसाइट्स एंड द स्टडी ऑफ ह्यूमन क्रोमोसोम्स, identification of the chromosomes and the construction of karyotype, observing the bending pattern through Giemsa staining. Now we are coming towards the materials and supplies, which includes 37°C incubator, centrifuge, refrigerator, tabletop with speed 200 to 3000 rpm, vortex mixer, laminar flow, microscope, slides, cover slips, bottle with lid, 15 ml falcon tube with racks, pasture pipettes, blood collecting, sodium heparin tube, syringes, tissue paper, micropipette, disposable gloves, surgical masks, complete culture media, colcimide, methyl alcohol, glycical acetic acid, aceto, organic stain, Giemsa stain, Sorensen's buffer, Na2HPO4, potassium chloride, KH2PO4, distilled water, 10% sodium hypochlorite, and plastic trays. Now we are coming towards day 1. Fresh blood is collected in a sterile sodium heparin tube. During this exercise, 2 volunteers or class fellows from the participants will be requested to donate 50 ml blood with syringe. The Qaid University Islamabad medical centers very near to the Department of Animal Sciences can help in this regard. Label the tube containing blood and carefully bring it to the lab. Clean and dry incubate with 70% ethanol and set it in operations. Make necessary adjustments at 37°C temperature. Clean the laminar flow cabinet and switch on the UV lamp for sterilization. Arrange the following items properly in laminar flow: culture media 15 ml, falcon tube racks, syringes 5 ml, disposable gloves, tissue paper, beakers 500 ml, 250 ml, and scissors in a plastic tray. The participants in the experiments could be split into 5 to 6 groups, A to F. First, instructors will show how open the culture media bottle and how to handle it up to culture in 15 ml falcon tube. We will use 8 ml media to culture the lymphocytes. To start the culture, take 0.5 ml blood from the sodium heparin tube and put it in the culture tubes. 6 proposed groups for conducting the experiments. These are group 1 male blood, group B female blood, group C male blood, group D female blood, group E male blood, and group F female blood. We have done this for shuffling. Mix the blood with the culture media with our lid and immediately incubate the 6 tubes in the present preset 37°C incubator. Note the time of incubation and then temperature of incubator and mix blood without the lid of the tube. Note: always wear lab coats and always use disposable gloves or handling other hazardous chemicals. All chemicals are hazardous. Never suck any chemicals by mouth. Always use micropipette tips. Decontaminate. Use stuff with 10% sodium hypochlorite, which we use in different labs. Day 2. All the participants should observe the culture tube and continuously monitor the temperature of the incubator. Shake the tube gently in order to mix the cells. While observing the culture tube, immediately close the door of the incubator, otherwise temperature will fall. In the evening, the participants should again check the culture temperature and again mix the cells. Preparations: hypertonic solution of 0.075 molar and Sorensen's buffer. For hypertonic solution, we will add 2.8 gram potassium chloride in 500 ml distilled water. Store at room temperature. While in Sorensen's buffer, 2 2 liters of distilled water, add 18.96 sodium hypophosphate Na2HPO4 and 1.6 gram of K2PO4. Day 3 morning, check the culture tube, mix the culture, not the culture of the media. Evening, again the check the temperature of the incubator and mix the cells. While day 4 prepare the fixative methyl alcohol and glycolic acetic acid in 3:1 at the beginning of the day 4 and keep in refrigerator until use. At the 70th hour of the culture, add 100 microliter chemical. After 72 hours, start harvesting the lymphocytes. While procedures in step 1, centrifuge all the 6 tubes at 1000 rpm for 8 minutes and remove the supernatant. Only 0.5 ml of the media is left in the bottom with pellet. While step 2, resuspend the cells in the remaining nutrients and carefully add medium and carefully add 2 ml of 0.75 ml molar potassium chloride drop by drop. Use the vortex mix machine for initiating. Add additional 8 ml 0.075 molar potassium chloride. While step 3, incubate for 7 minutes at 37 degrees Celsius and centrifuge for 8 minutes at 1000 rpm. Remove the supernatant and posture pipettes without disturbing the cell pellet. Step 4, resuspend the cells and fix them by adding 10 ml freshly prepared cold fixative. Firstly add dropwise and then initiate by vortex mixer. Now we are coming towards step 5, centrifuge at 1000 rpm for 8 minutes and remove the supernatant using posture pipettes. Step 6, repeat the fixation process 2 to 3 times. And on step 7, after all the centrifugation, resuspend the cells in small amount of fixative. Drop suspensions on a clean cold slide, air dry slide, and cover the slide with paper to avoid rust deposits. Now we are coming towards 8th step. Stain one slide from each tube. Observe the slide for chromosome in 20x, 40x, and 100x. Keep unstained slides in slide box in dry place for Giemsa staining. For Giemsa staining, stock solution, dissolve 3.8 gram of Giemsa stain in 250 ml of methanol. Warm the solution up to 60 degrees Celsius. Slowly add 250 ml of glycerin and filter the solution. The solution needs to stand a period of time 2 months prior to use. For working solution, add 10 ml of stock solution to 80 ml of distilled water and 10 ml of methanol. Giemsa stain is used on blood and bone marrow. Erythrocytes stain pink, platelets light pink, leukocyte nuclear chromosome stains magenta, lymphocyte cytoplasm stains sky blue. Thanks.